All right, guys, so we're going to take a look at topic 6, free response group B. And this one is a classic one. This is from a more recent AP test from a couple of years back. This deals with, again, multiple components within a free response. And this is dealing with mutations and looking at gene sequences and all that. So we have this whole thing with this Loeys-Dietz syndrome, right? And they set you up. Make sure you read the information they give you. This gives you a lot of information, okay? So again, we have this gene, okay? We know that this gene is part of a signaling process, and the gene makes a 565 amino acid protein with all these different structural regions, okay? And if we look at this picture, we can see that there's a signaling molecule binding region, so this is coding for a part of the protein that's outside the cell membrane. There's a transmembrane region, which is right here, right? And that anchors it into the membrane. And then we have this kinase region, right? And what does this protein kinase region do? It's involved in signaling inside the cell, right? And they tell you that protein kinases, they transfer phosphate groups to ATP to target proteins. And this is what it looks like. Okay, and then in the figure, always read the little chart in the figure because this really gives you some information, the explanation in the figure. Genomic structure, there are 7 exons, right? Labeled 1 through 7. And then the introns are the thin lines. Those are the non-coding sections. They're also giving you the start and stop codons, okay? And the portions of the gene that encode the different regions of the protein are shown. There's also a mutation. It's indicated by that arrow right off of box 5, M425V. What does that mean? That means that methionine 425, right, is substituted for valine, okay? Or substituted by valine. So that's a methionine gets substituted out and a valine takes its place, okay? So then they gave you these questions and they had this genetic chart, right, the code chart. So as soon as you see that, you know you're going to need to use that for something. But then they ask you about a pedigree. So this makes you get a little nervous because you're thinking, oh, Mendelian genetics, but relax. Okay, so you have a pedigree of a family. One parent in the first generation has the syndrome, okay, and a researcher claims that the disorder is dominant. So what would that pedigree look like? What would be a characteristic that would support the researcher's claim that it's dominant? And if you remember, autosomal means it's on a regular chromosome, so it's not on an X chromosome, which means it's not sex-linked. So autosomal means that it's going to be inherited equally, right? It doesn't matter if you're male or female. You have equal chances of getting it, okay? And then to make it dominant, okay, if you remember, we talked about in genetics, little people, big world, achondroplasia, right? Achondroplastic dwarfism is a dominant genetic disorder, okay? Individuals that have this are dominant. So if they have it, if a parent has it in the first generation, they can pass it on to their offspring, right? If the offspring inherits the dominant trait, will they express it? And the answer is yes, right? Because it's dominant. It's always being expressed. So if the offspring don't have the trait, they won't express the disease, which means they are homozygous recessive for it, okay? And so let's take a look at what the answer key said here for this. Let's see, here we go, okay? And so the answer here says, yep, okay, autosomal, inherited equally by males and females. And if it's dominant, it means that the phenotype most likely observed in each generation is going to be the dominant one. If it's not observed in one generation, you're not going to see it in subsequent generations. So if you have offspring and none of them have the disease, no one's ever going to get it again because it's dominant. So if it's not expressed, it means you just don't have the gene, okay? What this also means is that there's no such thing as being an unaffected carrier, okay? This is the same thing as Huntington's disease and Marfan syndrome, okay? All right, so then they want you to calculate the minimum number of nucleotides required in the coding region of the mRNA to produce and terminate the 565 amino acid polypeptide. So they gave you a hint here, to produce and terminate the sequence. So what does that tell you? To produce and terminate. All right, so 565 amino acid polypeptide. What is that coded by? Okay, it's coded by the DNA and the RNA. Okay, so nucleotides code for a polypeptide. Each amino acid is coded for by a codon. So how many codons are going to code for this particular amino acid sequence? 565 codons. Okay, so if you have 565 codons, how many bases are in each codon? There are 3. So you're going to calculate 565 times 3. That will tell you how many bases code for that amino acid sequence, okay? But they want more than just that. They want to know, what do you need to terminate it? So that means you have to add a stop codon, add 3 more to that, and you also have to produce it. So to produce it, you need a start codon, you need a promoter. So you have to add another codon. So really, that 565 amino acid polypeptide is coded for by 567 codons. Start codon, gene, stop codon. Okay, and then you also have to think about the number of bases. So you multiply by 3. So if you have 565 amino acids, multiply that by 3 nucleotides, get you to 1695. Now that earns you a point. They really wanted to see that you understood the concept of the start and the stop codon. And that's where these other 2 responses come in. Okay. Notice 698, you're adding 3 more. And then 1701, you're adding another 3. Okay. This final column, this includes the idea of the start and the stop codons in the production of the polypeptides. Okay. All right. Moving on to the next one. It says here, the question is, one genetic change associated with a disorder that results in a methionine to valine substitution is substitution at 425. You get a mutation. So what is the point mutation that would result from this? Okay, so your point mutation that you're going to get from this, okay, is going to be simply switching it from one base to the next. So what you're going to look at here is you're going to go from A to G and T to C, okay, or you're going to go AUG to GUG. Okay. Oh, and I skipped a part. Up here, the reasoning for why you see differences in the RNA. RNA is going to be modified or processed. So you're going to see a change in the RNA sequence from the original gene. We're going to get rid of the introns and put together the exons. We're also going to do some prepping of the RNA. We're going to add a poly A tail, and we're going to also add the methyl cap, right? And that's going to prevent it, the RNA from being destroyed, okay? We also might have some regulatory sequences in there that we might take out, okay, like promoters, enhancers, etc. Okay. Okay, and then the last part, 3 features of a model to connect a mutation with the activity of the pathway. So any 3 of these mutations change the structure of the polypeptide. If you change the sequence, it's going to affect how its signaling activates. Okay, changing the sequence is going to either trigger signaling or decrease it. Okay, if you change the protein, you're going to affect other things like what happens with the cell's growth, how does the cell respond to the environment, what kind of activity is going on within the cell that permits it to respond appropriately, what's the signaling cascade going to look like. Okay, so this question really covers quite a bit, and it even throws in a little bit of an understanding of what happens with cell signaling. So something like this would be a really good question on the AP test. Okay, all right, and then moving along to the last question on this one, they gave you this picture of a eukaryotic cell. Okay, this figure represents the process of expression of gene X in a eukaryote. So they want to know, okay, here we have our DNA, we have a primary transcript, and we go to mRNA and a protein. So they want to know what happens to the transcript. The primary transcript is 15 kilobases long. The mature transcript is 7. So this is 15, this is 7. So they want to know what modification resulted in the difference in length of the mature mRNA, and where does that happen. So you can kind of see in the picture, where is this happening within the cell? Okay, it's happening within a specific structure in the cell. Okay, it's happening in the nucleus. And why do we see a difference from the primary transcript to the mRNA? Right. If you're thinking spliceosomes and splicing, that's exactly what's happening. Okay, so let's take a look at that question, and you'll see here that what we're doing is we're removing the introns. We're simply doing RNA processing. You could say splicing is occurring, and this is happening in the nucleus. And then the second question was predict the length of the mature X mRNA if the full-length gene is introduced and expressed in a prokaryote. So if you take that same DNA, put it in a prokaryote, they want to know, what will the mature RNA look like? Will it be the same? Will it be the same as this? Or will it be different? And the answer is it's going to be different, right? What's it going to look like? Okay. it's going to be the same size. It's going to be 15 kilobases long. Why? Because mRNA processing only happens in eukaryotes. We do not see spliceosomes and the removal of introns in prokaryotes because prokaryotes don't have introns. Okay? All right, so that's it. That's it for this question. 4 points, 2 points for part A, 2 points for part B, and then 10 points on on question 1. Okay? That's it. Any questions, reach out to us. Good luck, guys. Keep plugging away. We're almost there. We're really getting to the end. So don't give up. Don't get frustrated. We're almost there. Okay. We're excited to be at the end. We know you are too. You've worked real hard. We've given you guys a lot of stuff and we appreciate it. Okay. We'll see you at the next video. All right. Bye, guys.